So today what we're going to do is we're going to talk about what's known as complex genetics. And this is kind of this next step after we talk about Mendel. So when Mendel was doing his work, he actually got very lucky with his pea plants because there was just dominant traits and there were just recessive traits. Every trait had two alleles. It was either tall or short. It was purple or white. It was either round or wrinkled pods or like seeds. There was no middle ground. He was very easily able to show that the idea that most people had of that fact that traits blended, like you had a tall plant and a short plant as a parent, that the children would be medium size, he was able to disprove that. He was able to show them that they were wrong. However, as people started looking into Mendel's work after he died, they started to realize there was more to just dominant and recessive. Now, we call what Mendel did either Mendelian genetics, which was the name of your notes, or complete dominance, means that they're either dominant or recessive. But there is more variation. And sometimes you can end up with multiple phenotypes, meaning you can have multiple things you see based on the genes in an organism. So these are what the notes are going to cover. We're going to talk about different types of inheritance or different ways that genes can be inherited and how it's different from what Mendel saw. Alright, so to start with, the first case is what we call incomplete dominance. This is really where what people thought of the blending idea, where you have a blue and a white flower, and you mate them, you end up with a light blue flower. So this is incomplete dominance. It's where the heterozygous one, the one that has a big letter and a little letter, is somewhere in between the two homozygous. And that neither allele is dominant. So, what happens is we distinguish the genotypes, the letters in the allele, by having one as the normal two capital letters and then the other one with an apostrophe. So, here's your example. Let's say we had a purple flower. So, big A was purple. The other one was white. So, we'd do A apostrophe would be white, and then the one in the middle, so light purple, would be one of each. So you'd have a big A and then a big A with an apostrophe. Not quite sure why they do it like this, but that's how they show it. They showed that they both are homozygous. You get the same allele for the purple and the white, but that they're equally, that neither one is completely dominant. The other one's not going to mask over it. So they equally contribute to the new trait. So that's why you get that mixture for the heterozygous. And we call them hybrids because they really are a mixture of the two. So an example, you have a red flower and you cross it with a white flower. So in this case, we're going to use R for red. So red is going to be two capital R's. White's the other end, so it's going to be two capital R's with an apostrophe on them. Okay? And then if we're going to have the pink flowers, well, the pink flowers are your heterozygous, so that means it's going to have one R and then an R with an apostrophe. So if you look at the picture here, you got a red flower, which is here. You have a white flower here. And when you get them, you end up with pink as their children. Alright, another example. If the dragons in Harry Potter have the fire power allele F for strong fire and F apostrophe for no fire that follow incomplete dominance, what are the phenotypes for the following dragon fire genotypes? Alright, so this follows similar to what we did with the previous genotypes. You have two big ones, you have a big and a little, and then two littles. Well, instead of having a little one this time, you have the apostrophe. So, two big F's, okay, so big F is strong fire. F apostrophe is no fire. Okay, so those are our two extremes. Well, then our heterozygous, one of each, is going to be somewhere in the middle. Moderate fire, middle fire, medium fire, however you want to put it, it's going to be somewhere between no fire and strong fire. Okay, so in incomplete dominance, neither one is dominant, and the heterozygous, the one that has one of each type, is going to be somewhere in the middle, or it's going to be a mixture or a blend of the two. Okay, so that's the first type of different inheritance. The second one is what we call codominance. This one is where you have two different alleles that are both expressed equally, or fully, I should say. So, our example here, we have a black and white chicken. We have a black chicken and we have a white chicken. So, we're going to use capital B for our black chicken, two capital B's. We use a capital W for our white chicken. So in this case, instead of a mixture, or excuse me, instead of a blend, you're going to get a mixture, okay? So you're going to have some white, some black, and it's going to look more checkered. Not necessarily spotty, but like checkered, or speckled is sometimes how it is called. So both the black and white are being expressed. You're seeing both the black and white. So the way that this is the heterozygous version, okay? So remember, BW is heterozygous. That is, you're going to get one letter from one type of genotype and the other letter from another one. Okay, so let's look at a different example. If mer people have tail color alleles, B for blue and G for green, that follow the codominance inheritance rules, what are the possible genotypes and phenotypes? Alright, so let's just go with the easy ones. So B, capital B will be a blue tail. Okay, they tell us that. Capital G is green. Good, they tell us that. Again, they tell us that, excuse me. Now, if we're going to get one of both, a B and a G, you're going to end up with blue and green tails. Okay, again, it goes back to the chicken. You're going to have, like, if these are scales, you're going to have some blue, some green scales. So it's not a mixture like a bluish green, like with incomplete dominance. It's, you will have blue and green. You see both of them in the codominance, like you saw both black and white. So codominance, you see both things show up, whereas in incomplete dominance, you see a blend or a situation that's between the two. This one, you see both of them. That's why it's called codominance. All right, next, a more specific example of codominance is called multiple alleles. All right, and this is where more than two alleles, so more than two things, control the phenotype. And the example we're going to use is blood type. So when you're doing blood typing, you either have type A, type B, type AB, or type O blood. So you have four types. So again, you have type A, type B, type AB, and type O. And the way that we recognize or determine the blood type is on your red blood cells whether you have a protein or not. Alright, and we have two proteins. We have protein A, we call antigen A, and protein B, which is antigen B. Now, you can have one, either, both, or none. So that's where we get the multiple alleles. You get multiple things coming into play. Now, the way that this is written, and this is very important, which I've included this in your notes, and this is something you need to know, is the types and the genotypes. So if you have type A, what does that look like on the alleles? The way that this is written is you have a capital I for the homozygous with the type of blood as an exponent. And if it's a recessive, meaning that you don't have it, so the capital I means that you have the protein. So this is the have protein. A lowercase I means there is no protein. Okay, so that means it's a recessive. Okay, so you need to know these. Make sure you star this. You need to know these. Okay, so type A, you can have two versions of it. You can have capital I raised to the capital A power twice. So this is your homozygous. Your heterozygous for A would be capital I, capital A with a little I. The same thing goes for B, but instead of an A, you have a B. You have a capital I, capital B, capital I raised to a capital B, that's your homozygous. Heterozygous would be a capital I raised to a capital B with a little I. Now, AB, there's only one version. You have to have one of each. So you have a capital I raised to the capital A, capital I raised to the capital B. And then O is neither. That means you have no proteins. Okay? So that means it's both little i's. Now, interestingly enough, O is considered the universal donor because what happens when you get this blood is your body will recognize these proteins. So if, let's say, you have type A blood, you can take type O blood because there's nothing on them. So your body it will be fine with it. But as a type O person, you can't take type A blood because your body's looking for cells that have no proteins, and then all of a sudden your body has proteins on it with these cells with proteins, and it will attack it. It will actually reject the blood, and it's a bad thing. So type O can donate to anybody, but they only can take O. And again, it deals with the type of protein you have. And I understand that the notation or the way we write it is a little weird, and I will try to explain a little better in a class, but let's look at an example. Hopefully that will help. All right, so you have a parent who has type A. In this case, they're heterozygous, so it's capital I raised to the capital A with a little I, and then you have a homozygous B. Okay, so you have two capital B's as the exponents. So we're going to do a normal monohybrid like we did before. So this is one box. So we're going to put the type A on top, that parent on top, and then the other parent on the left. Okay, and this is just like we did before, the Punnett squares. You take, you take the A and the B, so that gives you capital I, capital A, capital I, capital B. Okay, so you're going to do that twice. And then the other one is capital I, capital B, little I. All right, so that means 50% for the genotypes, 50% are going to be the AB here, and 50% are going to be the B. Okay, so when we look at this in terms of our chart, that means that these here are AB blood, whereas these here are going to be B blood. Okay, and again, you look at what letters are the exponents. Okay, the I is just telling you that it's blood typing. The exponents really tell you what type of blood you have, whether it's A, AB, or B, or if it's O, if you have the little i's. All right, our last thing that we're going to look at is sex-linked. Sex-linked traits are any traits that are being passed on the sex chromosome. Remember, the sex chromosome is the 23rd pair of chromosomes, and it's typically found on the X chromosome, because we're going to talk about in a second. Y doesn't really have anything we care about. Now, remembering from back when we talked about meiosis, this is back in November, we said that females are two X's. Okay, so you have an X and an X. Okay, so females are XX. They typically don't show a trait. They will be what we call a carrier. And when we talk about a trait, that means something, typically this is related to diseases, so this can be a bad thing, like color blindness. And a carrier means that they can give trait to children. Okay, so that means that, like, if you're colorblind, okay, so a mom can carry a colorblind trait, but she won't show it because there's two X's. One X will cancel the other because typically these traits are recessive. These that deal with illness. Males, on the other hand, are XY. Okay, so you have one X, one Y. That's a male, and the Ys don't carry any alleles that we really are worried about. So the X, which they get from their mother, is going to be what they have. So they're either homozygous dominant or homozygous recessive. They can't be carriers, and carriers are heterozygous because they're going to have one of each. And I just want to emphasize that. Carriers are heterozygous. So either a male, like if we're going color blindness, either a guy is going to either not be colorblind, will be normal vision, or will be colorblind. He has nothing to kind of buffer. He doesn't have that other X like females do to kind of offset the recessive trait that's being passed on. All right, so let's look at an example. The other thing is getting into the inheritance. All males will pass their alleles onto their daughter. Okay, so if a dad will always pass his stuff on to his daughter, But the females have a 50-50 chance because there's two X's. So here's an example. Hemophilia. That means you can't clot your blood. It means you basically bleed. You have normal, which we show with a capital H, and hemophilia, which we show with a little h. And the way that we express these, it's kind of like blood type, where we have a letter that we use as the base, and then you have the exponent as for the trait. We use the sex chromosomes. So a female, in this case, we have a carrier female, so she has a big H and a little h, and we put those as exponents on the XX to show that it's female. And then we have a normal male. So we don't put anything on the Y. It's only the X that we care about. So he has a capital H. So we do our little grid here. And when we look at this, okay, so this female has 2 capital letters, which means she's normal. This female has a capital letter and a lowercase letter, but remember, the capital letter is dominant, so it's going to be normal. This male has a capital letter. He just has that one. Whereas this one has a lowercase letter, so he's going to be a hemophilia. He's going to have hemophilia. Okay, so this is what I mean by the guys either have it or they don't. So in this case, the guy is normal. In the bottom case, he has hemophilia. And so if we look at it overall in the children, we can say, well, 3 out of the 4 boxes, that's 75%, are normal, and 1 box, 25%, is hemophilia. The other way we can say it is that 100% are going to be normal females, 50% are normal males, and 50% are going to be hemophilic males, which means they're going to have hemophilia. So you can look at it in terms of all the children, as in 75 and 25, or you can look at it in terms of, the females versus each other and the males versus each other. Okay, let's look at one more example here. All right, so in fruit flies, eye color is sex-linked. That means the color of the eyes is on the sex chromosomes. Red eyes are dominant to white eyes. If a red-eyed male is mated with a white-eyed female, then what percentage of the F1 generation will have white eyes? All right, so we're using capital R for red, lowercase r for white, because red is dominant over white. So our female, because she's because she's white-eyed, she's going to be 2 little letters. Okay, so X raised to the little r, X raised to the little r. Male is red-eyed. So we have an X raised to the capital R and the Y. All right, so we plug those in. We're going to put the female on the top, the male on the left. It doesn't matter which one you do. You can flip them as long as you put one on one side and one on the other. All right, then we match them up. So our first female is going to be X raised to the big R, X to the little r. So that means she's going to be red. Our second female is the same thing. So she's going to be red. Well, with the males, because they're getting their X's from the mom, you're going to have only 1 little letter. So that means that the males are both going to be white. So if we look at it overall, 50% of the children are going to have white eyes, or 100% of the males will have white eyes. All right. So the other thing that comes with all of these different types of inheritance that are different from what Mendel saw is that they're still only dealing with 1 gene. We have something known as a polygenic gene, which is where you have something that is produced by more than 1 gene. And so in this case, you get a continuum. So examples are your height, the weight, your skin color, your eye color, your hair color. So things like that, where you have multiple alleles, like I think for your eyes, I think it's 3 different genes. Excuse me, you have more than 1 gene. You have like 3 genes for eye color. That's where you get the continuum. Like you can have, it's not just brown and blue. You're gonna have light brown, medium brown. You have different shades of these colors, and we call that polygenic traits. Also, your genes are not the only thing that affect how an organism shows its traits, okay? So the phenotype is actually a combination of the genotype, what's in their gene, and the environment they're raised in. And if you've ever taken psychology, or if you ever take psychology, you'll hear about the nature versus nurture argument. Nature would be your genes, so nature is the genes, like what's in your DNA, and the nurture is your environment, like how is based on where you're raised affecting how you turn out. So an example here is for sea turtles. Okay, so if you have eggs that are fertilized in cold water, you're going to get males. However, if the eggs are fertilized in warm water, then you're going to get females. So the babies depend on what type of water they're fertilized in. Okay, another example is hydrangea flowers. Okay, so you can range from like a bluish violet to pink, and this depends on the pH of the solution. Remember at the beginning of the year we talked about pH. This is, the 1 through 14 scale. If it's really, really acidic, like if it's at a 4 here, you're going to have a really, really blue color. However, if it's more closer to neutral, then you're going to get pink. So it depends on the soil, like what the pH of the soil is. So again, this affects the color of the flower. Like the genes tell it one thing, but then the environment changes that or alters that. Okay, so it's not just the genes that are going to control the phenotype. The environment does play some roles in it. All right, so we're going to now do some practice problems with these type of inheritance. It's a wide range of different things, but it's the inheritance patterns are just a little bit different, but like the monohybrid and dihybrid crosses still follow the same rules. So you've got a little bit in terms of the phenotypes, you got some changes.